I'm really grateful to this invitation to speak and share some of our findings with this group of friends, really, in carotenoid research, addicts in this interesting and dynamic area right now. And also grateful to Dr. Howard and the supporters of macular carotenoid research, which are so important to us progressing and to having these opportunities to talk, because a lot of our research direction has been infused by ideas of people that you see here in the room. So today, I'd like to address the large number of environmental and genetic determinants of macular pigment density and risk for age-related macular degeneration, which provide clues about possible mechanisms to increase macular pigment and in whom increasing macular pigment might be most possible. And this is important because we will be learning over the next few days of the ways that macular pigment might enhance vision, but also might lower the risk for age-related macular degeneration, which you'll hear much more about. But just briefly, age-related macular degeneration in the developing world is an important cause of vision loss in older people, as well as the #1 cause of blindness when it reaches these late stages. About 20% of Americans over 65 have medium drusen that we can detect, or even an ophthalmologist can detect visually, which places them at risk for further developing advanced stages, which involve the loss of cone photoreceptors and the loss of vision where the cone receptors operate in the middle of the field of vision, as well as loss of ability to read fine detail. And this slide, the classic slide that Max produced, shows that the macular pigment, which accumulates in that cone-rich area, especially absorbs blue light, which is one mechanism by which it's thought to protect against macular degeneration. But I will also point out that there are some new data suggesting that in the more rod-rich area towards the periphery, one can observe some of the earlier changes in relation to age-related maculopathy. And there's the question about the possible importance of macular pigment in this more peripheral area. Some people may not have sufficient macular pigment to protect against this process. There is mounting evidence that macular pigment protects against macular degeneration, as we were talking about, and also serves possibly as a marker for other factors that do. And this was initially proposed almost 20 years ago by Max Snodderly, who noticed the kinds of correlates of low macular pigment were also risk factors for age-related macular degeneration, things like blue eyes and smoking and female gender. And this has been elaborated on by Stephen Beatty and many others, and supported by some cross-sectional studies. That is that of the landmark study of Richard Bone and John Landrum, which showed that the macular pigment in donor eyes, with macular degeneration was lower in those without. And later, we also showed in a large sample that I'll be talking about today that if you look at the women with most stable diets and the younger woman, that there is nearly a 2-fold lower odds for early AMD in people who have high macular pigment. The data that I'm going to be showing you today come from the Carotenoids in Age-Related Eye Disease study, and it is an ancillary study of the Women's Health Initiative, and we selected women from 3 of the 40 sites nationwide, and further selected women who had extremes in lutein intake. So it was 50% of women who had high and low intakes of lutein, so that we could have more statistical power to study relationships of lutein status to macular degeneration and other eye diseases. And we got the data that I'll show you today between 2001 and 2004, when women were between 55 and 84 years of age, and we collected macular pigment optical density measurements, extensive information about diet, supplements, as well as fundus photography to assess the presence and the severity of age-related macular degeneration. The Women's Health Initiative has also collected a lot of biospecimens, which have allowed us to measure levels of carotenoids as well as other nutrients, lipids, and inflammatory markers in the sample as well. And they have collected annually information on lifestyles, medication use, and chronic diseases, which you will see are some of the data that I'll report on today. And most recently, they've given us DNA, so we've been able to genotype for many of the genes, tag SNPs, many of the genes that are thought to be involved in the uptake of lutein, the transport, and the accumulation. And this is the machine. It looks quite different than the one Richard just showed us. This is the Macular Metrics 1 customized heterochromatic flicker photometer that we used. And this is really a dinosaur today, but really it was really quite sufficient and quite usable in getting macular pigment data in about 1,800 women. And at this time, they participated fully in getting measurements not only at one site, but actually 4 spatial locations, quarter degree, half a degree, 1 degree, and 1.7, compared to a reference of 7, in both right and left eyes. So this is, it's very reliable, and it's also very doable. Now, of course, there are clinical models that are now updated, and they're simpler, and they're more sometimes relevant, giving output for patients that might be relevant to treatment or their physicians. But it's really basically the same technology. So as you would expect, we found, of course, women in this study who ate more lutein and zeaxanthin in their diet and their supplements had higher levels of macular pigment density. But you can also see that on average, this relationship plateaued at about 3 milligrams per day. Now, these are average values, and I know that now many supplementation studies have shown that individuals can increase their macular pigment and have no plateaus that have even been noticed, or plateaus differ quite a bit between people. So I just wanted to point out the difference between average population-based data versus what you might experience in a clinical setting. But if you look at these as a scatter plot with individual points, you can see there's quite a bit of variation around that basic relationship of increase of macular pigment density with increased lutein intake. So that many people with low intakes have high macular pigment, and many people with high intakes have low macular pigment. So the considerable amount of variation. And you might think, well, this might be due to inaccuracy of dietary information or differences in ability to absorb carotenoids. But when you see, we've done this as well for serum carotenoids in people with stable diets, that we see very much similar relationship. A lot of scatter about the expected relationship between increasing levels in the serum and density of macular pigment. So why do some people with high lutein intake or blood levels have low macular pigment, while others with low intake or blood levels have high macular pigment? And why does the level of response vary so widely? Now, within samples, there have been anywhere between half of subjects showing response of macular pigment response to dietary intake, up to 95%, probably somewhat the composition of the sample or of the supplement matters. But also, even within samples that have a pretty high response, there's quite a bit of individual variation in the level of response. And so I'm hoping that as we understand the determinants, genetic and environmental, we can understand some of the reasons for this varying response. And also possibly predict who might and who might not respond, not only for clinical use, but also for use in clinical trials. If clinical trials need to be short, if we can predict who might respond, we might be able to include more people in a study to understand our results with a little less variability. So we're going to talk about dietary, health, lifestyle, and genetic factors related to macular pigment, which include other aspects of diet and nutritional status, and conditions which influence the uptake into the gut or to the eye, and also conditions that are related to metabolic syndrome, inflammation, and oxidative stress. When I talk about the genetic factors, I'm going to talk about common polymorphisms, common variants related within genes that are related to, we think, based on other evidence, the ability to take up, accumulate, and metabolize carotenoids. And I won't be giving a lot of detail. I won't be saying the full name because I want to give you an overview picture of what is related, but to go to more detail, you can see our paper that was published earlier this year. Nutritional attributes, there are 4 nutritional attributes which appear to enhance macular carotenoids. One are those which influence beta-carotene metabolism. Another which influences another influence on macular pigment is the intake of high fiber foods, the intake of fat, and the intake of certain types of fat. One of the strongest genetic determinants of macular pigment in this sample was the presence of certain variants in this BCMO gene, and this stands for beta-carotene monooxygenase 1. This gene protein cleaves the beta-carotene to form 2 vitamin A molecules. And George Leitz has found that SNPs that are associated with the SNPs that we've studied are associated with higher activity of this reaction, resulting in more conversion of beta-carotene to vitamin A and lower serum levels of beta-carotene and higher lutein levels. So the higher lutein levels in the serum might be a reason for finding such a strong association between beta-carotene monooxygenase variant and macular pigment. And also recently in the ARIDS trial, beta-carotene in the supplements was found associated with lower lutein. So there are a couple studies that have found that there might be competition between beta-carotene and lutein, which results in different ability to accumulate macular pigment or have an effect. So here are the data. You can see that I've listed here macular pigment density by tertile of lutein intake, but also by the number of G and A alleles for this particular SNP. And you can see that if you look at individual genotypes, so people that have a GG genotype, for example, as dietary intake increases, so does macular pigment density. See the same for people with 1 A allele and 2 A alleles. And within each level of dietary intake, you can see that with each additional A allele, we have higher average mean macular pigment. And what's interesting is right here, you can see that women who had 2 A alleles had a macular pigment density which was near to the value of women who had dietary intakes of much higher levels. So the AA allele for this particular SNP, for example, could be an indicator of people who have good response to dietary lutein. We found also that women with low intakes of fiber-rich foods had low macular pigment density levels. Why is this? Well, fiber is a marker for whole fruits, whole vegetables, which are high in antioxidants and anti-inflammatories, anti-inflammatory substances. And also, those types of foods are related to lower glycemic response, and that might be related, as we'll talk about a little bit later. We also found that women with low intake of polyunsaturated fat had lower macular pigment density. And this is consistent with data in other studies that show when a full-fat salad dressing is added to a salad, we absorb carotenoids better than if a fat-free dressing is used. Or another experiment showing when a high-fat spread is given with lutein, lutein absorption is higher. Or when eggs are eaten, even though eggs have a small amount of macular pigment, because of the lipid matrices that they're in, there is a higher macular pigment response relative to the level of lutein. So we need a little bit of fat to get the lutein in. We also found that women who have high intake of omega-3 fats, either from foods or from supplements, had higher macular pigment density. And also, women who had certain genotypes. Now, these are genotype, particular polymorphisms within genes which encode proteins that enable us to convert medium chain omega-3 fatty acids, such as alpha-linolenic acid, to long chain fatty acids, which we need, like DHA and EPA, by the eye for a variety of reasons, including the structural integrity of the retina. And so women who had genotypes, which are also in other studies associated with high omega-3 levels in the blood, also had higher macular pigment density. Another common trend we found is that women whose blood lipids or genotypes were related to higher HDLs, which we've heard carry half of the macular carotenoids, had higher macular pigment. Here, serum triglyceride, I'm using as a surrogate, high serum triglyceride as a surrogate for low HDLs. But you see that if you have women who had low HDLs, had low triglycerides correlated to high HDLs, had higher macular pigment. And these other 4 genotypes have also been shown in other studies to be related to higher levels of HDLs or higher stability of HDLs, and they too were associated with higher macular pigment. And I'll take time to describe one of these as an example. The ABCA1 gene encodes a protein which is an ApoA protein, which is on HDLs, and it's important for the stability of HDL molecules. In chickens who have mutations in this gene, they develop white legs, they have white blood, because they have an inability to distribute carotenoids, even though they take it in the body. There's a similar human condition, Tangier's disease, which is a result of mutations in this particular lesion, which provides good evidence that stability of HDLs is important for the accumulation of carotenoids in various tissues. In addition to those genotypes, John talked a little bit about GSTP1 that Paul Bernstein's group identified as the zeaxanthin binding protein. We found that one SNP within this protein was associated with higher macular pigment. We don't know if it's the SNP associated with higher levels of this protein yet, but we'll be interested in knowing if that's the case. And then we found that macular pigment is lower in women with one or more conditions that are associated with metabolic syndrome, that is, conditions like hyperinsulinemia, central adiposity, blood lipid abnormalities, or hypertension that generally increase the risk for diabetes and cardiovascular disease, or oxidative stress or inflammation. All of these conditions are thought to be associated with higher risk for macular degeneration, as well as many other chronic diseases of aging. We and many others have shown that people who are obese have lower macular pigment density levels at all levels of dietary intake compared to those who are lean. We observed that women who had diabetes had lower macular pigment density levels at all dietary intake levels relative to those who did not. And here I show you not only those relationships, but also other conditions associated with metabolic syndrome. And you can see that in all cases, people that have these conditions associated with metabolic syndrome have lower macular pigment, including having high triglycerides, having hypertension, or having 2 ApoE4 alleles. Now, you look at many of these bar graphs, and you go, well, that's not a very big difference in macular pigment. Can that matter very much? And when we're looking at individual conditions associated with lower macular pigment in a sea of many that can influence variability in macular pigment, it's like looking for a needle in a haystack. They're hard to see, and so that we're even seeing any, and all of these are significant associations, so seeing any even significant associations, I think, are important. But also, if you look at the joint associations, women who have 5 of these conditions compared to none have 2-fold lower macular pigment density. So while you might see these are small on an individual basis, when you add up into similar processes, they might make a marked difference. Oops. We also found that macular pigment density was lower in people who had other conditions associated with inflammation. Now, also, obesity and diabetes are conditions which are often associated with higher levels of inflammatory markers. We also measured C-reactive protein in those who had high levels, which is a marker of having systemic inflammation, had lower macular pigment density. Also, having low vitamin D is now being recognized as a potential indicator of inflammation, in addition to status for vitamin D. It decreases, for example, after surgery. And we found that people that had low levels of vitamin D had lower macular pigment. And again, when grouping inflammatory conditions, we find a larger effect. I told you that most of these are being measured. I think I maybe forgot to tell you that these are all from macular pigment density that we measured at ½ a degree from the foveal center. And almost all of the associations that I've shown you are similar, regardless of what eccentricity we look at. Here's an exception. We didn't see a significant association for complement factor H. You probably all know that complement factor H, Y402H allele, is a risk factor and a very important risk factor for macular degeneration, resulting in higher levels of complement factor H and a more exaggerated inflammatory response. And you can see that women who had, we did these analyses a little bit different, and we have to look a little deeper in order to see some associations. We only looked in women who had above average lutein intake. And we asked who among these people have uncharacteristically low macular pigment. And you can see that women who had low macular pigment despite high intake are more likely to have the risk alleles for complement factor H than those who did not. And we found macular pigment was lower in women with many conditions associated with oxidative stress, such as lower intake of vitamin C, one of the most powerful water-soluble antioxidants in the diet, and lower levels of vitamin E in the diet, one of the most powerful lipid-soluble antioxidants. We also found a strong association with lens density, which there's evidence that lens density is an indicator of lifetime oxidative stress. It's associated with telomere length, leukocyte telomere length. So that is a pretty strongly developing rationale for a possible oxidative basis in relation to macular pigment, as well as to variants in genes that when mutated are associated with high levels of oxidative stress. I won't go into that right now because I know we're short of time. But when we add up several of the conditions that are associated with oxidative stress, we find a stronger association most of the time than when we look at individual factors. I also mentioned, many of you know that we didn't find, like many of you did, that smokers had lower macular pigment density in this sample. And there were only 2% of women in our sample who currently smoked. But we did look a little more deeply, like we did with complement factor H. And we found that women smoking more than 7 years did have low macular pigment despite high lutein intake. So it was there, but we had to tease it a little bit to understand it. And smoking, of course, creates oxidative stress. So putting it all together, we have found that macular pigment density, the conditions favoring higher macular pigment density are, of course, the intake of their constituent parts, luteins and zeaxanthins, fruit, vegetable, and grain-rich diets, high-fiber diets, high antioxidants in the diet, having some lipid, as well as having either omega-3 fatty acids in the diet or supplements, and or the ability to convert medium-chain omega-3s to long-chain. And we found that higher macular pigment was favored in people that had a genotype that is related to beta-carotene converting to vitamin A, which generally results in lower blood levels of beta-carotene and higher levels of lutein. We found that macular pigment was higher in people who had genotypes related to having either higher HDLs or higher genotypes related to HDLs and the ability to transport and export cholesterol. We found that higher macular pigment was common in people with some variations in the gene which encodes the protein that binds zeaxanthin in the eye. And then we found there were many factors generally associated with chronic disease that were associated with low macular pigment, including metabolic syndrome, inflammation, and oxidative stress. I decided not to talk with you about some of our other data today, but there are several genes, when mutated, lead to maculopathies. And we found that, and that's a very rare event, but we found common variants in these same genes were related to lower macular pigment. So interrupting the retinal integrity by a variety of biochemical mechanisms was associated possibly with lower macular pigment. Putting together with this with AMD risk factors, we found that, first of all, many of the genotypes which are related to lower macular pigment in the study were also related to AMD, and that it will be published soon, or hopefully it's under review. And I put a red box around all the factors associated with macular pigment, which are also risk factors for AMD. And you can see that, as Max thought almost 20 years ago, the story keeps continuing to unfold that risk factors for AMD are associated with lower macular pigment. So a concluding hypothesis is this. Macular pigment density might be an integrated marker of macular carotenoid intake, but not only that, also conditions which favor their uptake, accumulation, and retention. So we're not going to know much about a person's status if we simply ask them how much lutein they take in. But having integrated marker can be useful. But also, it might be a risk marker for age-related macular degeneration. And if this is true, it could be that measuring macular pigment at early ages, before even early macular pathology is developed, could be a risk indicator for propensity to develop age-related macular degeneration or similar diseases. So thank you for your time. I thank many collaborators on the project and point out that many people here have contributed to this research. This work that we do was really stimulated by Max Snodderly, and Liz Johnson has been a contributor for over a decade in this research too, made possible by the flicker photometer made by Billy Wooten. Randy Hammond, who's helped us adapt a protocol which is repeatable and usable over many different sites. And Paul Bernstein, who has collaborated in understanding some of our genotyping data in relation to AMD. Thank you. Any questions?
There's one question there.
Thanks, Julie. That's really cool data. And I have a bit of a question about statistically what it means to characterize these data. Mm-hmm. So it looked like there were occasionally very large, occasionally very small, but meaningful differences in all of these categories between macular pigment density and sort of a specific genetic marker or the presence of some condition. So I know statistically I always worry about things like family-wise error, multiple comparisons of the exact same thing over and over again. And I don't think I'm really clear on genetically what constitutes a family. Is it sort of one genotype and then multiple alleles are measured and that's sort of a quote, family? Or is it, do you have to do something like a Bonferroni correction for every single one of those markers? And if you've done that, does it influence the statistical significance you were seeing?
Well, the issue you speak about is really important in genome-wide association studies, but it's less relevant here. Clearly, it's true that we're looking at many associations, and we actually didn't expect to see so many associations. But we have other ways of seeing whether we have false positives. And one way is to see if you consistently see the association by different lutein intake strata, by different waist circumference strata. So you'll see in the paper we published earlier this year that we have consistency across those levels, so we don't think that they're chance associations.
You showed that vitamin C and vitamin E gives rise to lower levels give rise to lower MPUD levels. You can imagine that it's just because having a low diet, and if you take the lutein intake into account, do these rations still hold?
Yeah, I should have said that everything I showed you was adjusting for lutein intake.
So what would be the mechanism that vitamin E correlates with a low, and vitamin C with a low macular pigment optical density?
Well, I would say propensity for higher oxidative stress, either systemically, which could lower, maybe increase the turnover of carotenoids. Do the chemists agree with that? Or higher oxidative stress in tissues due to lower concentrations of those antioxidants. So that I would presume that that would be possible.
Okay, thanks.
Mm-hmm.
Just one final question.
The data on ABCA1 polymorphism is very interesting, and fit with the one chicken with ABCA1 loss of function completely. Have you compared patient with the same HDL cholesterol level due to ABCA1 mutation or to metabolic syndrome, because this HDL level may not be originating through the same mechanism.
I'm sorry, I didn't
Yeah,
understand.
you can have low HDL because you have ABCA1 mutation, for example, or because you have metabolic syndrome,
Yes.
and they are not the same, because in
Yes.
one case ABCA1 is functional, not the other one. So if you try to compare lutein level in these situations.
I can tell you that the genotypes I showed you are independent, adjusting for all other genotypes. And however, some of the factors that I put in the slide weren't in those particular analyses. Oh, wait, no, actually, actually triglycerides was. So in most cases, these are independent associations, and that's why they're so small. I think that there is some possibility for overlap, and what we're doing right now is some more complex statistical modeling to understand the joint associations. When you pile them up on top of each other and you see a stronger effect, you're less worried about the overlap. But I think it's a good question because we did a lot of data, and what we're doing right now is really trying to sort out just the kind of thing that you're saying. Which of these are saying the same thing, and which of these are contributing new information? We just simply didn't expect to get so much new information to have to sort out. But I think it's interesting and important what you suggest.
Yeah, we did some studies on people with ABCA1 mutation and metabolic syndrome, and there are some differences. We can discuss it. Yeah.
Yeah, I think that, clinically could probably add some insight to this. Mm-hmm.